Hello everyone, my name is Dagmara Błaszczyk and today I would like to present you my project, which topic is ViralCounter, an automated algorithm that allows to count the number of viruses in cells. Currently, we are living in a pandemic's world, so there's nothing strange about the number of projects related to a coronavirus. So it's mine. My project is about the usage of image analysis techniques on the images of cells that were infected by coronavirus. Usually, those kind of analysis is performed by scientists in laboratories via some existing tools like, for example, ImageJ, but it is a quite time-consuming process. So, as the topic of my presentation says, the goal of my research is to create an algorithm that will count the number of viruses in cells with the focus on its location. So that means that we want to count the number of viruses inside the cell and also outside the cell, very close to its border. And as I've mentioned before, for scientists in laboratories is a time-consuming process, so I hope it will facilitate their work. Our dataset contains 68 3D RGB images which were infected by COVID-19. This dataset was provided by Virogenetics group from Małopolska Center of Biotechnology, and those images are in a very specific for fluorescent microscopy format. In those images, we've got three different components marked in three different colors. In red, we've got lectins in cytoskeleton, which shows the borders of cells in an image. In green, we've got viruses, and in blue, we've got nuclei of cells, so we know how many cells do we have at one 3D image. The first step of our analysis was simple pre-processing, like for example the removal of the noise from an images, but the second step was the determination of the area of clearly visible cells in a 3D image. So for that purpose, the first thing that we did was the determination of separated objects on each layer of the image. And we could obtain three different types of objects. Usually all of those types of objects are present in one 3D image. And depending on the type of objects, we needed to perform various processes which led to obtain the data set with clearly visible cells extracted from an image. And in this step, the most important information was carried out by blue and red channel. The three different types of objects that we can find in an image are objects with one separate cell. So here the area of the cell is already determined and those type of objects were automatically extracted from an image. The second type are objects with few cells which borders are visible. So here cells are usually connected by a small piece of them. So that means that we can still separate them correctly. And for that purpose, I created a function that separated cells in the narrowest point. So here we extract only the cells that were connected by a small part of them. The ones that were connected by a large part of them are excluded from our analysis. And the last type of objects are objects that are densely condensed. So that means that there's a lot of cells inside these objects and they cannot be in an easy way separated. And also in this class of objects, we've gotten objects that are localized at the edges of images. And they were just excluded from our analysis because they don't provide us the full information about viruses in cells and also its location. And after the extraction of the cells from an image, we needed to remove the remaining parts of other cells, which usually are close in the image, and the remaining parts of those cells were localized at the edges of images. And the whole algorithm was performed through all the layers of the images. So that means that the size of an image with one cell extracted from the full 3D image was determined by the layer at which this cell was the biggest. As a result, we obtained a data set with 3D RGB images of cells extracted from 3D images with a bunch of cells by an algorithm. The figure 3a shows the layer of 3D image, and the figure 3b, c, d, and e are the chosen cells by an algorithm. Usually the one 3D image have approximately 20 cells, so our algorithm is able to extract few up to 10 cells. It depends on how they are distributed in the image and also how many different types of objects do we have in one image. And then the data set will be used to train an artificial neural network for a segmentation of areas of viruses inside and outside the cell. And in the last step, we will just count the number of segmented objects from the images from the neural network. Here I would like to thank to all of my colleagues that are involved in this project, and also thank you for your attention.